Hi, this is the second part of a two-part series that I prepared for the Year 9s that I teach, the iGCSE Biology Cambridge examination. And for anyone that's joining us, this is pretty much what I would expect iGCSE biology students to be understanding by the end of their first year of GCSE Year 9. So we're going to go through this assessment. It's made up of around about 10 multiple choice questions and 20 marks worth of open response questions. So it starts off with a very quick one. This is nutrition related. Which reagent is used to test for reducing sugars? The answer was Benedict's. The building blocks of protein molecules are amino acids, and the nutrient which helps to make hemoglobin, which transports oxygen in the blood, is iron. Nitrogen is the element found in all proteins, but not in carbohydrates or fats. The active site is the part of an enzyme molecule into which a substrate molecule fits. Why does the rate of an enzyme catalyst reaction increase as temperature is raised? The answer is that the molecules actually have more kinetic energy stored inside them, so they collide more frequently, and the chances of successful collisions are increased as a result as well. There's a big crossover with chemistry as well. We start to teach collision theory at the end of year 8, and those people who have also done chemistry as an iGCSE, one of the last topics in the year was collision theory as well. Changes in pH do affect the shape of the active site. That's why changes in acids causes great sensitivity in enzymes. Enzymes are very sensitive to pH. They only work in a specific range because the hydrogen ions can latch onto different parts of the enzyme, and the active site's shape changes. Correct order for alimentary canal is esophagus, stomach, small intestine. Muscular contractions that move food through the alimentary canal is peristalsis, and bile salts help with digestion because they break up the large droplets of fats into small ones, linked with chemistry because it increases the surface area. That process is called emulsification. Canines are for piercing and tearing the food. You can talk about ripping, pulling food apart, that's absolutely fine. Molars are for chewing and for grinding food. Can't say cutting or biting because that's kind of like vague, and also cutting is the kind of job of the incisors really. So the key to the next one was making sure that there are very, very clear comparisons between the two different skulls and the two different types of teeth or absence of teeth that exist between a tiger skull and a rabbit skull. So the tiger, you can see that there are more pointed incisors here. The rabbit doesn't have such pointed incisors, or they're rather less pointed. The tiger actually has canines there as well. You can actually see these canines just on this side, but the rabbit doesn't seem to have that. The tiger has jagged premolars and molars. You can see the jagged edges there, whereas the rabbit's premolars and molars are flat. And the tiger has fewer molars when you look at it compared to all of the molars that the rabbit has. You can talk about that too as a comparison. The rabbit has a diastema. This is actually a large gap between the incisors and the premolars. Tigers don't have that gap. There's no gap, or rather, you could say that there's a small gap between the incisors and the premolars. So that's a diastema. Now, the giveaway with the teeth is that the tiger has canines. Okay, that indicates that the tiger is a carnivore, and all these jagged premolars and molars. But you could also look at other evidence in the skull. The eyes are positioned at the front of the skull for the tiger. Also, there's a pointed ridge, and there's a pointed crest on the skull as well. That could relate to all sorts of things. For example, aerodynamicity of the predator. You need to have a low pH in the stomach because that's the optimum pH for the protease in stomach acid to break down proteins. Villi are adapted for absorption. The most common answer for this was a large surface area, but you can also talk about the fact that it's got lacteals on the inside as well, good blood supply, very thin walled. You've got microvilli as well that actually increase the surface area even further. The test to show that proteins are present in a liquid is the Biuret test. Violet is a blue color, so if it's added to a sample that does have protein, it turns from blue to lilac. I did accept purple. Lilac, though, is it seems to be the most commonly agreed shade and word to give to describe the purple color of any sample that Violet is added that contains proteins. Next question. Well, why enzymes really necessary? Well, just the simple fact of the matter is that without enzymes, you have processes that just take too long a time. Enzymes also make reactions much easier to take place because of the fact that they lower the activation energy or the energy required for a chemical reaction. The reactions that enzymes enable in the body would require a higher temperature in order to take place, and it's just not feasible for organisms to raise their temperature to that level. And also the nice thing is that enzymes are catalysts. They speed up the chemical reactions without taking part, being used up by the actual process itself. So they're very specific as well. So with enzymes, you could also suggest that there's like an alternative pathway energetically. It is actually the liver that makes bile in the next question. The gallbladder is actually the very tiny sac that that stores it. Kind of took me out earlier this year. The gallbladder is about a fingernail's worth, and with the wrong kind of diet, it crystallizes. You get crystals. The wrong type of cholesterol can actually cause extreme pain and needs to be taken out. Yes, so the organ that stores bile is the gallbladder, and the component of a balanced diet which is necessary for the correct formation of feces is, of course, fiber. You could obviously also give the term roughage, and that concludes the biology, the second biology assessment. If you've joined from the outside, thank you very much for watching. Feel free to take these questions. They are past paper questions from Cambridge example. You can adapt to use your own initiative, come up with your own quizzes, or you can put it into a suitable piece of software. I hope it's useful, as well as the thoughts that I've shared with you about where students actually go wrong with this one. And if you're one of my Year 9s, thank you very much. You now need to go to your dirt reflection form, and you need to fill that in for this assessment. Thanks for watching. Stay safe. Bye for now.